Hi everyone, you are on module 44 right now. As you must have already seen the task list, in module 44 we are going to go through some multiple choice quiz questions on enzymes, and we'll also do the end of the chapter questions. So we are now wrapping up unit 3, and hence I thought that reviewing the whole unit and looking back at what we have learned about enzymes is very important right now. So why should we review? The reason is that we might get bored by the term review, but it's a very important study skills, as normally a human mind tends to remember things that are recently taught. But then after a gap of a couple of weeks, you will again start forgetting what you have learned about what is being taught now. So right now that we are closing the chapter enzymes, we should do a quick recap of what objectives we have already studied from this unit. So let's begin. So I started reviewing the whole unit through a mind map, and I thought I could take you through the mind map step by step. You could also pause the video and continue doing your mind map while going through this video. Or you could, towards the end, I've asked you some questions, just work on that. But make sure that you also review along with me. If you find the video slow at any point, please feel free to watch it at a 1.2 or 1.75 speed. Just increase the speed of the video. So I begin with the first learning point or objective of this unit, that is what are enzymes and location of enzymes, enzyme structure, mechanism of enzyme action, factors affecting enzyme activity, inhibitors that prevent enzyme from working, uses of enzymes. So if we start with what are enzymes, you can see I have highlighted the keywords biocatalyst, required in very small amount, and they can be reused by the cells. Most importantly, they lower the activation energy of the reactions that they catalyze. The second objective or the second important learning point from this unit was about the structure of enzymes. They're mostly globular proteins. You can also see that I have highlighted in red that some enzymes are specially not proteins. They are made up of RNA molecules and they are called ribozymes. Enzymes possess a specific region called active site, which has a 3D shape because of the tertiary folding of the protein, and that is because of a specific sequence of amino acids in the polypeptide chain. So if you give excess heat, this tertiary structure gets denatured and the enzyme stops functioning. Next, we will focus on mechanism of action, where we studied two theories. One is the lock and key theory, and the other is the induced fit model. The induced fit model is the most accepted one because it states that the substrate induces the active site to rearrange itself so that the substrate and the enzyme complementarily fit to the most best fit in the three-dimensional space. Why? Because substrate and enzyme are always in a state of random motion due to their kinetic energy, and when they successfully collide, means just when the substrate collides with the active site of the enzyme, they form a temporary complex called the enzyme substrate complex. Immediately the substrate is then converted to product either by breaking down or joining of substrates from the product. Once the product is formed, it leaves the active site and lets the enzyme become free to catalyze the next reaction. So this entire description comes under how the enzyme works or the mechanism. The lock and key hypothesis was earlier the most accepted hypothesis, which said that the active site is completely complementary to the substrate, and only one substrate can fit the enzyme. But later on, it was found that some enzymes can be complementary to a group of substrates which have similar structure, which can induce the shape of the active site. So induced fit theory became more accepted than lock and key theory. Coming to enzyme locations, we studied that there are two categories of enzymes, intracellular and extracellular. Intracellular enzymes are produced by the cell itself through protein synthesis, and then they work in catalyzing reactions inside the cytoplasm, like catalase. Here I have also given two examples of extracellular enzymes, that is amylase and trypsin. Extracellular enzymes will be produced inside the cell but secreted by the cell to the outside to do their function, to do their catalysis. Also, a key highlight on certain enzyme kinetics, that is, when enzyme catalyzes a reaction, how does the reaction proceed? So the initial rate of reaction is always very high, but once all the active sites of the enzymes are saturated, the reaction reaches a maximum velocity, means that is the highest speed at which the reaction will proceed, because all the active sites get saturated or filled with substrate, and new substrates being added have to queue up and wait. So at that point we say that the reaction has reached its Vmax or maximum velocity. If we calculate the half Vmax and extrapolate that point with the curve, we get the substrate concentration at which half maximum velocity is obtained, and that is called the Km, or the Michaelis constant. Can you recall what does the Michaelis constant or the Km value tell about the enzyme? If you remember that, pause the video and quickly jot it down in your notebook. Later on, you can add that to your comment. The question is, what does Km tell us about an enzyme? The Km value. Okay, moving on to the next part of our mind map, or next concept that we had learned in this unit, is all the factors. Very important, because most of the questions related to graph interpretation comes from this area. We studied about how temperature increases the kinetic energy of the enzyme and the substrate so that they collide even more frequently, and successful collision means more enzyme substrate complex formation and rate of reaction increases. But every enzyme has a specific temperature at which it catalyzes the most, that is the optimum temperature. Beyond that temperature, the enzyme begins to denature. The next factor was pH. Similarly, every enzyme have their specific pH at which the 3D shape is perfectly complementary for the substrate to come and bind. And if you change that pH, if you decrease that pH or you increase pH beyond the optimum, the 3D structure of the enzyme gets affected because of high or low hydrogen ion. So yes, every enzyme has an optimum pH at which they will catalyze the maximum, or the rate of reaction becomes maximum. The third factor is enzyme concentration. That is, if the concentration of the enzyme increases, that means you're adding more enzyme molecules to your solution mixture or reaction mixture, and more active site means there will be more substrates binding and more product can be formed per unit time. That is, the rate of reaction will increase. You can explain this using the term more enzyme substrate complex formation. Then the fourth factor is substrate concentration. If you keep on increasing the substrate concentration as the reaction proceeds, and keep the enzyme concentration fixed or definite, definitely there will be a point when all the active sites of the enzyme will get saturated, or they will get busy converting helping the substrate form product. So that's when Vmax is reached. So substrate concentration increases, yes, rate of reaction will increase, but up to a certain point. Beyond that point, the substrates have to wait till the active sites of the enzymes are free. So I just briefly mentioned that, and I have highlighted some important terms while putting them on the concept map. Then I thought we should add some important points about two types of inhibition, reversible and irreversible. I remembered that toxins and heavy metals make permanent bond with the active sites or allosteric sites of the enzyme, and they affect the 3D structure of the enzyme. Whereas some reversible type of inhibition is done by competitive inhibitors and non-competitive inhibitors. I remembered how the graph looks like, so I wrote how Km value and Vmax value get affected with these two types of inhibitors. But I did not add a lot of details because I want you to look at this part of the mind map and think of what more should be added to make this extensive mind map more detailed. Think about what is the structure of a competitive inhibitor, where it binds. Similarly, what is the structure of non-competitive inhibitor, where does it bind with the enzyme, and how does it affect the enzyme's activity? And you can add them in different branches. Then I was also thinking about the industrial and the laboratory purpose of enzymes in the commercial world right now, and how industries and laboratories are using the enzymes to its best possible way without wasting it. So that brings us to the idea of enzyme immobilization. What are its advantages and disadvantages? So after the end of this video, I want you to work on enzyme inhibitor and uses of enzyme, these two aspects of this mind map. How can you enhance your mind map and add more details to this? You can work on only these two parts of the mind map, or you can actually work on the entire mind map by yourself now, because this run through would have helped you a lot in recollecting all the objectives. After this, you will post your mind map in the comment section. That's all that I wanted to discuss. Just take you through the whole unit and reflect on what you have learned about enzymes till now. Thank you, and please share your mind maps or the part of the mind map that I asked you to focus on in the comment.